All right, so part 2 of our cell membrane lecture. We've already talked about the cell membrane and that the current accepted model of the cell membrane is the fluid mosaic model. Fluid because the components are not cemented into place, so they have some ability to move and shift within the membrane. Mosaic because of the fact that the cell membrane is made up of a lot of different types of components. So we've got proteins, we've got glycoproteins, glycolipids, phospholipids. There's a lot of stuff that makes up that membrane. We also mentioned the fact that the cell membrane is a phospholipid bilayer. Bilayer, bi means two, so there are 2 layers of lipids. Here's one layer, there's another layer, and then they are oriented so that their hydrophobic tails face in and their hydrophilic heads face towards the watery environment. So this is facing toward the outside of the cell. These hydrophilic heads are facing toward the inside of the cell. The cytoplasm is a more watery environment. So how do things move across that cell membrane? Diffusion is the key. If this represents a membrane like the cell membrane, molecules are going to diffuse from an area of higher concentration to an area of lower concentration. So in this image, you can see there's lots of molecules of dye here, but none on the right side. Because of the fact that this membrane is selectively permeable, it has little gaps in it, over time, some of these molecules can pass through. The thermal energy that these molecules have due to motion, that gives them the movement that they need to spontaneously and without work, they're going to move and spread out until they come to a dynamic equilibrium. So this would represent a dynamic equilibrium because these little molecules, they're still moving. And so some are still moving this way and some are still moving that way, but the net concentration is equal on both sides. Now this is in absence of any external force. So this is something that's going to happen on its own naturally without any external force applying any sort of pressure to it. So a substance is going to move spontaneously within its own concentration gradient from an area of higher concentration to an area of lower concentration. And it's independent of any other stuff. So these molecules of dye, they're going to move along their concentration gradient according to molecules of dye. If we had molecules of dye and salt in here, the salt is going to move along its concentration gradient. The dye is going to move along its concentration gradient, and that's going to be separate of each other. They're not going to work together. So simple diffusion, we're moving from a high concentration to a low concentration. Electrochemical gradient is a term that you're probably going to hear a bunch. Electro indicates ions move from a higher concentration of ions to a lower concentration. Chemical is just talking about that, solute concentration gradient. So it can either be ions or it can be other concentrations of other substances. But the gradient means that they're going from high to low concentration. All right, so what does this mean for our cell membrane and moving across the membrane? The cell membrane is selectively permeable. It controls what moves in and out. It allows some substances to cross more easily than others. The things that are small and nonpolar, they can diffuse very easily across this membrane, even through these little phospholipids. They can just squeak through here. So that would be things like oxygen or carbon dioxide, even small water molecules and very small lipids. They can just kind of push their way and eke their way through the membrane between the phospholipids. But not everything can do that. The characteristics that prevent molecules from being able to squeak through those little phospholipids are size and polarity. So once again, I mentioned that really small things can move through here, oxygen and carbon dioxide, small lipids. Oxygen, carbon dioxide, those things are not polar, so they can also move through relatively easily. Not everything can move through as easily though, so sometimes it needs some help. Facilitated diffusion. Facilitate means to help something. So facilitated diffusion is going to help speed up the process. When I have my little CO2, O2, even my little water molecules squeaking through here, they're able to do that without any energy. The energy is not required. They're still naturally diffusing across that membrane. Facilitated diffusion is also without any energy required. We don't need energy to get them across. Facilitated diffusion just gives them a pathway so that they can diffuse more quickly across that membrane. So this embedded protein right here would represent a channel that helps to facilitate diffusion. It's a gap that's always open, so these little molecules can more quickly move across along their concentration gradient. Or if these molecules are larger, or if they're polar, then it allows them to move across the membrane along their concentration gradient. Notice they are still going from higher concentration to lower concentration, so that's the direction that they're moving. All right, so, oops, let me back up a step. So this is a protein right here. And this is a protein that's embedded within that cell membrane. So why are proteins perfect to be those kind of structures, to act as channels to facilitate diffusion? Well, the reason is because some proteins are polar, some proteins are non-polar, some proteins have both characteristics. So by building these proteins within the membrane, we have an area, the embedded area. This guy right here is actually made up of non-polar amino acids. They are hydrophobic, and so they like the hydrophobic tails of the phospholipids. And so this portion of the protein anchors the protein within the cell. Oops. These outer portions of the protein that stick out of the membrane, these guys right here, they're polar. They're made up of polar amino acids. And because they have polar amino acids, it makes this portion of the protein and this portion of the protein hydrophilic. So these guys are going to extend outside to the extracellular fluid or inside the cell into where the cytosol or cytoplasm is at. So because they have both of those components in this protein, they're going to embed within the membrane but still extend and be happy in the hydrophilic portions, both externally of the cell and internally of the cell. So an example of a protein channel, so as I mentioned before, water can easily pass through that membrane. It's kind of small, but it takes it a little bit of time. It's slow getting through that membrane. So this is a protein channel called an aquaporin. And aquaporin allows water to go through a lot faster. It's basically a big gap in that membrane, a big open door, so it allows it to pass through. Another example was our hydrogen proton pump. And so you can see the hydrophobic structure within the membrane and the hydrophilic structure. And this allows those hydrogen ions to move along their concentration gradient. So perfect example of electrochemical gradient that we're talking about here. So here's ions for the electro part, water for the chemical part because it's just a molecule or compound. And so both of these guys are going to move along their concentration gradient. So because the membrane is selectively permeable, it is specific about what it allows in and out. And those protein channels are also very specific. So the aquaporin, it only is specific to water. That's the only thing that it's going to allow to come in and out. These specific channels allow specific material across the membrane. The items can diffuse without help of a channel. They're very small, the O2, CO2, small lipids, so they can squeak by, but these channels allow them to move through faster. Most of the nutrients that we need within the cell, they need some sort of assistance across that membrane, so they're going to need some kind of channel to get across. Notice that some substances move into the cell and some substances move outside the cell. NH3 is ammonia, So it's one of the waste products that are going to be produced by cellular processes, so the cell wants to get it out. All right, so some of those proteins that act as channels, they're only going to open in the presence of a specific signal. Ion-gated and voltage-gated, these are basically, they're gates on the top of that protein channel. So it keeps that protein channel closed until the stimulus causes that little door or gate to open. So like ion-gated channels, the neurotransmitters, they're going to bind to the gate, and that allows that channel to open up and sodium can enter the cell. We'll see more about ion-gated channels when we talk about the nervous system. Voltage-gated channels, so once again, when we have a nerve impulse that's moving down an axon, it changes the voltage of that structure, and so changing the voltage can also cause those gates to pop open. Both of these are still passive. They do not require energy. Just when those ions are present, it causes it to open. When that voltage changes, it causes it to open. But this does not require any additional energy. This does not require any additional energy. So, getting through the cell membrane, we've talked about passive transport already. These don't require energy. Simple diffusion is just directly through that membrane. These are oxygen, carbon dioxide, little lipids, little nonpolar substances. They just squeak through that membrane between those phospholipids. Facilitated transport does not require energy, so it's like an aquaporin. It's going to help things move through faster. It's also going to provide a channel, provide a door into the membrane or out of the membrane for molecules that are larger, polar, hydrophilic, and like I said, it provides faster transport for any of the smaller molecules. Both of these are going to move along their concentration gradient. No energy required, but moves from high concentration to low concentration. Active transport. Active transport, this is diffusion against the concentration gradient. Because it's moving against what it naturally wants to do, it requires ATP, it requires energy, and then we need some sort of a pump to move it against its concentration gradient. So for active transport, it's going to need energy. Notice that these molecules need to be inside of the cell, but the concentration of yellow molecules are much higher inside the cell than outside the cell. So it's going to require energy to move from low concentration to high concentration. No energy required for simple diffusion or facilitated transport. Active transport requires energy. Let me flip back. Okay, so on this guy right here, these little yellow molecules, they are moving along their concentration gradient, and as they move, they're able to pull the little pink blocks in, which are moving against their concentration gradient. That's going to require energy. On this one, once again, the little yellow molecules, they're moving along their concentration gradient, but when they move in, that's allowing the little red molecules to move against its concentration gradient. Red was lower here and higher here, so it doesn't want to go from low to high, it wants to go from high to low. This little protein pump, these require energy, it requires ATP, in order to move those molecules against their concentration gradient. All right, so water, we already know it has some very interesting and very important properties. So the specific case of water moving across the membrane, we're going to look at individually.